So when we last left off, we're going over the different types of RNA. And I picked a few. Some of them should be a review. We may go into more detail than what you're used to or you've heard about them in the past. And then some may be new to you, right? And so in particular, we've already covered tRNA, which is the transfer RNA. Today, hopefully, we'll get through mRNA, rRNA, the micro and small interfering RNAs, small nuclear RNA, the small nucleolar RNA, and possibly pRNA. I looked at my notes for PV. I have a whole bunch of like little history about pRNA, but I left it behind in my office. And let's see. Okay. And so we talked about tRNA. Remember, it's given this a lot of times to draw the secondary structure as this clover leaf. In reality, it's like an L or a 7, the state of Florida, a gun, whatever your inclination is, but in 3 dimensions. I don't know. That's kind of like a Rorschach test, I guess. That's not the Rorschach blot. Sorry, not you. Those are ink blots that psychiatrists have you look at. Okay. Awesome. But we did go over the different parts of the tRNA, and it's easier to show it on this. Whoops, I'm going to go to the next one. It's easier to show on this than it is on in its tertiary structure. And so we have the acceptor stem, also known as the amino-acyl stem, the D loop, the anticodon of D loop and D stem, anticodon loop, anticodon stem, T-C loop, also known as the T loop, and the T-C stem, or T stem. Okay, and it's always 5' to 3'. But in 3 dimensions, it looks more like that, and that's why it's color-coded accordingly. Why does it fold? Thermodynamically, biophysically, that is the most stable structure. It's the way that they form these helices. Those helices line up almost like really two, because I can focus on the colored part, but almost like 2 really big helices, but in reality they don't, they're thin. Okay. And so, it's kind of like, but yeah, they just, and then they lock in place. So, and it's very, very stable. tRNAs are actually are more stable than a lot of the other RNAs because they are very, they have a very more rigid structure. It's still flexible, but there's still some variability in that variable loop, which is not shown on there. The variable loop kind of sticks out. The one that would be on this would be really small. Sticks out between the T-C stem and the anticodon stem. Okay, so the variable loop sometimes is right through here. Sometimes it's very, very small, like just like a little bulge. And so, which would be this part right here. And then for a couple of them, it's really extensive, like it sticks way out. So, pardon? Okay. We talked about the maturation pathway and how these are really ways to control how much tRNA is around, which is really important because if you control the tRNAs, you can also control protein expression. I should say expression, protein translation, protein synthesis, okay? And so one thing is, I forgot, someone on the exam mentioned something about how they alluded, I forgot how they worded it in the part about RNAs, but how essentially all RNAs were transcribed as mRNAs. That's not necessarily true. The small RNAs and like tRNAs and stuff in our RNAs are not considered mRNA. Okay, so that's why the primary transcript is not always mRNA. But the first step is the concomitant step, the one that commits it to the pathway, and it's one of the ones that can be easily regulated. Okay, that's the one that I worked on for my PhD. All right. And then this is to show you the fact that tRNAs are involved in protein synthesis. They don't make the proteins themselves, okay? But it's what brings in the amino acid. That's why it's called the amino-acyl stem. Okay, and the other end of the tRNA would actually contains the anticodon, okay, which reads, for lack of a better word, the mRNA's codon. Okay, and that occurs where? Where is the reading of the mRNA by the tRNA? Where does that occur at? Ribosome. All right.
If all of the tRNAs have the CCA
Uh-huh.
attaching to it,
Yep.
why
Oh, that's right. So what charges, it's not charging as in adding a charge to it, but charges the amino acid group actually also has to have that fidelity. And it has to be able to read, the enzyme that does that reads the anti-codon loop to the amino acid stem. It recognizes the structure. So it too, that's why it's very important. Any variation in the tRNA, especially in this region down here, will result in it not being the correct functionality for the tRNA. Okay, and just remember, there's also always going to have to be a D, which is a modified nucleotide here, and there's always this T psi C, which that's a pseudouracil, which is also another modified one. All tRNAs have to have those. They're modified, or otherwise they don't work either. Okay, tRNAs, if we talk about relative size of RNA, it's probably a good medium sized. Okay? They are not tiny like microRNAs, which are only like around 20 or so nucleotides long, but they are not as big as the next one, okay, which are mRNAs. This is the one that you guys are probably most accustomed to seeing and talking about. Literally, it just means it's a direct transcript of the DNA that contains the codons. They usually can be quite large, right? Because you can imagine if you have a protein that's 100 amino acids long, then just the gene, before you even take out the introns, stuff has to be at least 300 nucleotides. And you're going to have introns in it, and you're going to have the precursor part, you're going to have the poly A tail, you're going to have all these different parts to the mRNA, so they can be quite large.
Are they the biggest, or is RNA bigger?
It depends on the gene. There's certain genes that code, for example, fatty acid synthase in mammals and us. It is massive. I don't think that there's any RNA or any other RNA, any other RNA that can compete with it. I never emphasized that. So we'll actually talk about that next semester. I mean, it's a huge. It's so big you can actually see it on an electron microscope. So, and it's a single gene in humans, whereas in bacteria and stuff, it's actually multiple. Okay. Righty. I don't have any, because mRNA, there isn't like a common shape to it. Our RNA typically is large. There are some that are smaller, but typically we're talking about things that are large. One thing that's very special about our RNA is that it is a ribozyme. mRNA for us typically is not a ribozyme. I say that, does anyone know why I said it's specified for us, for humans? One? No. Because there are, especially viruses, that can make transcripts of RNA that act as ribozymes on themselves. They make what's called a RNA structure. Like hepatitis does that, mosaic virus, some of the other viruses, where they actually attack themselves to excise themselves out of the genome. We don't do that. Okay. But what does a ribozyme mean with respect to our RNA? Our RNA. Something like a tongue twister.
Ribose that acts like an enzyme?
It is an RNA that is an enzyme. And know that if you are RNA, ribosomal RNA, let me just say that. Ribosomal RNA truly is a ribozyme. Okay. In fact, it makes what's called a ribonucleoprotein complex. Do I not have that on there? I do not. Nucleoprotein, which you can figure out just based on that. Sometimes these are called RNPs because the ribosome itself requires both RNAs and proteins to come together in order for the chemistry to occur, but it's the RNA that contains the nucleotides that does the chemistry. So that's why it's a ribosome, because the RNA does the chemistry, but it's ribonucleoprotein complex, and it's huge as well. There are very few different types of RNA, ribosomal RNA. They're very specific. They have only one functionality for the most part, so there's not a lot of variation there. Whereas other RNAs, like mRNA, there's lots, right? Because every gene is going to have one. tRNAs, there's several. Because every amino acid has at least one or two. And I think there's at least one in the universal code that has 6 different tRNAs that can make it. So that's why there's, and know that they can be highly modified. All right. Let me bring this up. I really like this picture, and this is when I wish I could show it in 3 dimensions. That's really cool. There are 2 subunits, and you probably remember, like in bacteria, when you take genetics. Do you remember what they call the 2 subunits? 50S and and 30S. So we have 50S and 30S, and when they come together, it makes the 70S. Because cell biologists, they can add so well. But do you know what the S is for? That's almost like worth bonus points.
Structure?
No. It's a unit. No.
Substrate?
No. It's named after a person. That's why I said it's great. It's great. It's called a Svedborg.
Oh, yeah. Oh, yeah.
And to be honest, other than really old papers that talk about things, and with respect to some RNAs, the ribosome, the primary one, no one ever uses it. And no one ever talks about doing proteins based on. Now we use molecular weight, which molecular weight is linear in that sense. It doesn't change. Svedborg has to do with buoyancy. Oh, yeah. I don't know why it never stuck around historically. All I know is whenever I took a class at the school, we had to learn how to calculate Svedborgs, and it was a pain. But you had to do like an ultracentrifugation gradient, and they had to spin the protein or the RNA, whatever it is we're going to do, for really long periods of time, and the cesium chloride gradient, because cesium chloride is really dense. So what happens is you have to do it for like a day, for really long periods of time, and then they just see where it's at, and then measure it back, and see that there's Svedborg. So it has to do with shape, it has to do with buoyancy, it has to do with essentially its mass. I mean, there's lots of things that it takes into account. But that's why 50 and 30, but whenever they come together, they have a different shape. Like it's not truly additive, and so therefore it comes out 70. But so RNA is about the only thing. Once you hear about like the 5S RNA, which is a smaller subunit, I can't tell you the part. Smaller subunit of part of the RNA. But I don't know of anything else that really keeps around the spec ones. They just refuse to go back. It's like the U.S. with miles. Why do we keep miles around when everybody else is kilometers? Right? So what we do. Okay, but I like this because of the fact that you can see where tRNA binds. Because the tRNA are these brighter colors. Does anyone know what these darker colors are that's hanging off towards like the periphery? What is the ribosome made up of? rRNA, ribosomal RNA, and those, whatever those are. What else is in the ribosome? What kind of complex was it that I said earlier? Ribonucleoprotein? So what do you suppose those are? Nope, not nucleic acids, because this is nucleic acid, all those letter colors and stuff. Those are the proteins, and these are little tiny proteins that help hold everything in good place where they should be, but they don't do the chemistry. You can see they're not even located here at the active site.
What's the function of those proteins?
Some of them, I don't know what the functionality is for all of them. Some of them may be due to help the complex formation for the RNA structure for the ES and for the S come together. Sometimes, like for RNAsP, what the protein does is it helps, it doesn't do, because it's also a ribosome, it doesn't help do the chemistry, but it helps select for the substrate for the product. Because you can imagine, if you have a binding site here, it's going to be recognizing both the substrate and the product. If you can have some way to where you can only say only the substrate is allowed to come in, then it makes it more efficient. So it's a balancer. So, no, that's not very good. But what they've done here is they've broken it apart some way. You can see clearly you see 3. In some books, I didn't check most recent edition, but some books that only talk about 2 having 2 binding sites to the ribosome. Some will say 3. Okay, there's A, P, and E. And some do not consider E an actual binding site. Then some people argue between mammalian and bacterial, and they say, well, mammalian's clearly got a 3rd, whereas bacterial don't. When I learned that we learned 3, A, P, and E. Does you know what E is? It's where the first entry. First exit. So, okay, so, but yeah. But yeah, it's like, it kind of gets kicked out. We'll talk about it in a moment. Oh, but yeah, if you crack, oh, actually I got a better picture coming in. We can see the E site. We see the E site. A, B, E. So that's why I'm saying, so it's like, it doesn't have any chemistry, but there's a tRNA there.
Function is to mark extension.
I don't know, I mean, I don't know, some may argue, and this is me, some may argue by having that one there, you don't have to worry about going in reverse, right? It's an exit only kind of thing. I don't know if that's true or not. That maybe whatever the proteins that are around here versus the proteins right around there help keep it only going in one direction. Because you don't want it to go frontwards and backwards. There's no proofreading in that sense. The only proofreading part is between the things like codon and codon.
Are there any cases where it does go backwards?
Not that I know of. Otherwise you die. Something when I mean, you can stall. I'm sure it can get stalled. In fact, I know it can because you can make analogs that come in, and that's actually how they do all crystallography to know where these sites are. Is they do an analog that cannot be hydrolyzed, or cannot be amino-acylated, not peptidylated, amino-acylated, and so then you get stuck. And so they're like, oh, we got stuck in this position, and since it looked a lot like it. So sometimes instead of having a phosphorus, maybe it's a band-aid, because the name looks a lot like it, or sulfur, but or arsenic or something like that. But then the enzyme can't work, you get stuck. Okay. All right, how are we doing in time? Well, that's probably. So it's closed, so you can't see. It's like cloistered. The tRNAs are cloistered in there, but when you crack them open, you can see where they're bind. And so they really bind at that interface between the 30 and the 50 subunits. I think this is the 30, that was the 50. I don't know the label. That's why I said the eyeball, that's why I would say. Okay, but what you can't tell here, which is why I wish I could take it out and rotate it around, is you've got 3 tRNAs. And so what's happening is as one tRNA comes in, it's like little bumper cars. They kind of like hit along and then they little simply like birds. They come in, the next one comes in, next one comes in, and they leave. Okay, so that's what happens when you crack it open.
How would one crack one open?
Pardon?
How would one be able to crack it open?
They can take they've since crystallized this. I mean, that was a major feat. The people that have done this work won lots and lots and lots and lots of awards. Because this is huge. And I realize you guys don't know a lot about structure. You're just used to seeing structures like this, but there are limitations. So like NMR typically can only look at really small things, because otherwise the signals get too convoluted. X-ray crystallography can usually look at larger things, but there's still a size limitation to it. Electron microscopy can only look at big things, but there is no resolution. Like you can only see, oh, that looks like to me, they almost always look like a honeycomb cereal. But you know, it's like, oh, that looks like a honeycomb cereal. You can't see it, no, there's a penis there. So they all have limitations. So originally they can see it in electron microscopy, and then they X-ray crystallography got better and better and better. All right. Please start. Oh, right. So I don't know if I forgot to double check to make sure that all my links still work for the little movies. I apologize if they do not. But now there's lots of things out there that can show you this. But I would expect you to know this. Okay. So actually, you got cut off. So. There are 3 steps in general to this process: initiation, elongation, and termination. Yeah, it's not there, but it's termination. If I say IF and EF, it's because those are initiation factors that help get the thing started. Get the thing. That's so not smart sounding. To get the process started, EF are elongation factors that help keep it going. And what terminates it? A stop codon. That's the start codon. Close. But yeah, it's a stop codon. And what does the stop codon do? Does it code for a tRNA? Does the tRNA have a stop codon? Yes, it does. There is a tRNA. In fact, there's what? 3 stop codons. I used to all of them now. And they are tRNAs and they code for water. Water just comes in, kicks it all off. So, so what happens first is that the start codon is AUG, okay, and it always codes for, and this is a little simplification, it codes for methionine, but what kind of methionine? Does anyone know? It's not the normal. It's not just plain Jane methionine. It shows it as this, but sometimes they even give it a little special asterisk or something. What methionine does anyone know? It has an old-fashioned name. You know how acid, ACET, means 2? Well, there's one called formyl, FORMYL, which means 1. It's one it's in methionine, it's got an extra, it's got an aldehyde stuck to it. So it's kind of a formyl one, so it's called formyl methionine, or FMed. Sometimes there's a little case F there. Most proteins, though, after they're made, get post-transcriptionally modified, and that gets cleaved off. That's what gets the ball started. Okay, so we have the FMet, the formulated methionine, and then that's going to help recruit through the initiation factors and get the entire ribosome up there. Elongation factors is what helps keep it going process. Oh, and this is called, the first site is called the A site, and then the second site's P, and then like I said, one that's not here is the E. There is a little bit of, well, I don't want to say contention or debate. Like I said, some say that bacterial don't have it, some say it's only a million. But don't be surprised if you see pictures where there's one that says E for the exit, or that now have the empty tRNA that's kind of hanging out right here, still hanging around until it gets kicked off. All right, so what happens is there's translocation that occurs, and the methionine is now in the P site, which stands for peptidal transfer. Is that right? It's the peptidal spot. Okay, and this is the amino acylation spot. So what happens is now for the second codon, we have another tRNA that's going to code for that codon right there with its anticodon, and it's going to bring in that second one. It's not always a serine, but this is one here showing you that it's a serine. And what occurs within this is that the whatever amino acid and the growing peptide chain that's in the P site is going to be added over to the A site's amino acid. So now it's on that second tRNA that gets kicked off. Like I said, it may get shoved over to the E site, and now the growing nascent chain is in the P site for the third one to come in. And then what's going to happen is whenever the third one comes in, this is going to be added to the glutamate. And so then we'll have 3 hanging off of its tRNA, which then will be translocated over, kicks off the previous tRNA, and keeps on going on and on and on, until you bring in that water. And the water is the one that's acts as a nucleophile that comes in and smacks off the rolling chain. Okay, to make the protein. All right. And that's a lie. The movie is not available on the course website. Well, actually, I don't know. It may still be on the course website, but I haven't kept up that course website, so I don't know what else is all on that course website anymore. So that was before we had MyFire. I used to have one that was just a course website that had movies and stuff like that. Oh, are there any questions over that?
So the E site isn't necessarily a part of the ribosomal unit, it's just that space outside of it?
Well, so if we go back, let me go back to the walnut looking thing. What we're going to do is, the next class, because I'd like to take a lot of time for doing this, like I said, this is probably the only time that you ever cover these. Unless you take some of the special class courses that cover some specific ones. But we're going to pick up on Monday, we'll finish covering microRNAs. Hopefully remember to bring the PB notes. And we'll also probably start chapter 4 lipids. Lipids will usually take you as long. All right, so I'm going to go ahead.